Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So good morning, everybody. This was a wonderful workshop up to now. Thanks for coming to this session, Imaging in Science, a Big Data Challenge. We have three distinguished speakers in that session, and they will all talk on different aspects of at the cutting edge of statistical inference, efficient statistical inference, large scale or large data optimization, and certain aspects of physical science. In that case, the main applications are actually in biophysics. So to start with the first speaker, who is Timo Aspelmeier. He's going to explain a little bit of very exciting new statistical challenges arising actually from what is called nowadays nanoscopy or nanoscale fluorescence microscopy. As you maybe know, until the early 19th of the last century, the so-called AB limit stands for roughly about 100 years and tells you that you cannot discern object with visible light actually beyond half of the wavelength you use. And then in the early 90s, essentially Stefan Hell from the Max Planck Institute made a groundbreaking observation how it is possible to break that limit. And from that time on, a rapid development actually has been initiated. And nowadays, a lot of activities is going on in understanding and observing living cell and transport communication processes. For example, if you look to a recent issue of Nature Medicine, you find actually that about 80% of all papers are based on nanoscopic techniques. So consequently, the last year's Nobel Prize in Chemistry has been awarded to these three gentlemen, Eric Betzig, Stefan Hell and William Mörner, for their fundamental work. Interestingly, by education, they are all three physicists, but they got the Nobel Prize in Chemistry. Timo will now, what does this work? Here is an example from what is called stochastic marker switching microscopy, and actually the movie should work, but it doesn't. Let me see. Yeah, now it works. So what you observe is, and we will learn more about it in Timo's talk, a timely, highly resolved and spatially highly resolved response of photon counts, and from that you finally have to aggregate an image, which is in this case a part of the alpha tubuli network in the cell skeleton. This is what classical wide field microscopy gives you, and this is what nanoscale resolution microscopy gives you, and there are certain statistical and computational images we will learn about. Then Victor Panaretus is going to talk about a different aspect. Again, his applications are from biophysics. These are so-called DNA mini-circles, and he will talk about also spatial temporal issues, that is functional data analysis of spatial temporal time series, and here methods which come in are in particular certain kinds of spectral expansions in order to model that time dynamics. Finally, Günter Walter is addressing a detection issue. We learned about that already in the talk of Boaz Nadler yesterday. In a sense, detection, we know that of course statistically is simpler than estimation, than recovery of the whole function or image or whatever. And he will give us theory for precise detection boundaries, in particular in a multiscale setup and fashions. So I'm looking forward to all the three speakers. And our first speaker is Timo Aspelmeier from University of Göttingen. And I will just set up his slides now.
Good morning. So I would like to tell you in this talk about some statistical issues and challenges that arise in super-resolution microscopy. And this is joint work with Alexander Egner from the Laser Lab in Göttingen and Axel Munk. And first of all, I'd like to introduce you to this it's not complete at all. It's a list of today's microscopy methods and how far you can get in resolution with them. So wide field microscopy is sort of standard microscopy, which you probably all know. And then there are several methods, and some of them, STAT and SMS, are going down or are improving the resolution of a standard microscope by at least an order of magnitude. And that's, of course, an incredible gain, because then you can get down to scales of 10, 20 nanometers where you can see sort of individual proteins. And I'm going to focus on just these lower three of these methods, and I'll show them how they work, and so you can understand where the statistical issues come in. So before I start, I would like to introduce you to sort of a standard microscope, because there you can see where the problem actually comes from. The sort of resolution boundary, the diffraction limit, as it's called, has its origin in this picture. So on the left, you see some object, and that's being magnified by this microscope, and you look in from the right, and in fact, you can achieve very large magnification factors. That's not a problem. But magnification is not the same as resolution. So the main problem that appears is if you imagine a light source, a point source over here, it emits a spherical wave, and this wave enters the microscope and is transmitted to the other side, and it's focused onto one point. But it's not the entire spherical wave that enters the microscope. It's only a small part of it, the small part that goes into this opening angle there. And this is a fundamental loss of information. You cannot recover the rest of the spherical wave. It's lost. And that leads to the effect that on the other side, what you get is not actually a point. It is not focused on a point, but instead you get a blurred image like this. So you start out with a point source, and what you get is a blurred image of that point source. So this is the point spread function, as it's called. This is this green blob over here, and I've shown a cut through this point spread function, and you can actually calculate what it looks like. That's this green curve, and it's actually pretty complicated. It's got a single peak in the middle, and then some small peaks, and there are infinitely many to the sides as you go further out. And this point spread function has a certain characteristic width, and effectively, you cannot see any details which are smaller than this width of this point spread function. And this is the fundamental diffraction limit, and it was thought for more than 100 years that it cannot be broken because it's a fundamental thing. You lose information right at the start, and you cannot get it back. Before I'm going to tell how it can be broken anyway, I must tell you something about fluorescence molecules. Because in today's biological applications, this is what is being done, and it's actually crucial for these super resolution techniques. So what's a fluorescent molecule? It's a big molecule like this, and it has several electronic states, one ground state and a higher energy state and many more. And it can happen that if you use laser light of the correct wavelength, you can cause the electronic state to change from the low ground state to the first excited state. And then, after a short time, it relaxes, and then it falls down into the ground state again, and it emits a photon. But it's a photon of a slightly different wavelength than the one that you excited it with. So it's shifted to longer wavelengths. So this is very exciting because you can now attach these molecules to basically whatever you want. You can target a specific protein of interest, for example. Say I want to know something about that and how it works in a cell, and then you can make that, you can arrange for that protein to be labeled with these fluorescent markers. And then what you do is you shine the blue laser light onto them, excite them, and they emit a green light. So you make them light up by themselves. And that's a very exciting thing. These molecules have several other characteristics. One of them is unavoidable, basically, which is that they can break. So eventually, a molecule may sort of do this circle here many, many times, emit many photons in the process, but eventually, and that's a stochastic process, it will break. And then it's dead, and it can't emit any photons anymore. The other thing that's very important is that you can have molecules that have a dark state. So there's a molecule in this configuration, and it doesn't emit any light if it's in this configuration. But you can make it switch from there to the active configuration, again, by stimulating it with a laser light. So you can turn these things on or off, as the case is, and that will turn out to be crucial as well for what I'm going to talk about. So now we have an optical microscope, and we have fluorescent molecules, and this is what a fluorescence microscope looks like. You have a sample down here, and it consists of a set of markers, fluorescent markers. You excite them with this blue laser light, and they start emitting photons. These photons are transmitted through the microscope. You observe them on a camera here, and what you get is this image. It's still very blurred because you haven't done anything about the diffraction limit yet. So you observe this green cloud of light. Okay, so here's an example of that. This is an image of a hippocampal neuron, and here's an enlargement. And what you see is, the first thing you see is it's very blurred. That's the point spread function. There's also a lot of image noise, and the noise comes from various sources, actually. So the primary source is that we are actually counting single photons here, because the fluorescence molecules emit single photons, and that's what we are seeing, and that's what the camera is recording. So this usually gives rise to pulse or noise, but not always. And there can be very much more complicated forms of noise here. Then there's the camera at work, and that converts the photons into electrons. And then you have electron multiplication noise. That can be modeled by a gamma distribution, for example. Then you have some analog circuitry in the camera, so that adds some Gaussian, perhaps, noise as well. So all of these images have a very complicated noise structure. Okay. Yes?
Basically static the whole time you're doing the measurement? Like at the scale? Like, do they move?
You mean the markers here?
Yeah.
Well, they may move, and I'll show an example of that. But in principle, you would hope they are static. Yeah.
What's the variability sort of between the picture noise and the actual image that we're after? So like in the
well, if this is pulse or noise, I mean, it depends on the number of photons you count.
Right,
and
so what's sort of on the order of what are we counting?
Well, this is, say, 100 photons in bright spots, so it's really noisy. Yeah. So how do you actually break this resolution limit? And the first idea was, as Axel mentioned, by Stefan Hell. And what he did was he had this excitation laser. And the excitation laser is now focused on a little spot. This is the spot here. The whole thing is the blue excitation spot. And it's diffraction limited because you can't break it. You can't break this limit. But then on top of that, he applied a second laser, this red one, and he also focuses that, but in a special way. And so the red excitation field has actually a donut shape. So it's a ring with a hole in the middle. And it's got a different frequency. And what it does is it's working like in a laser, in fact. It stimulates the emission of photons. So if a fluorescence molecule is hit by this red laser, it thinks, OK, I must emit my photon. And it does so very quickly. And it does so with a different wavelength, the red one. But that means this molecule is effectively switched off because it cannot emit the green photon anymore. And that's called stimulated emission depletion microscopy. And so the effect is that this donut-shaped ring, it's also diffraction limited, so it doesn't have any sharp edges or anything, but it's got this hole in the middle, and if you superpose that with this blue excitation light, there is only a small spot in the middle from which the photons that you observe can actually originate. So whatever photons you see here on the other side, you know they must be coming from this little spot in the middle. And that's the gain in resolution that you can get. And then you do this for 1 pixel, and you scan the whole image, and then you get your total image. And that's really a very ingenious idea, I think.
When was that developed?
In 1994. This is a technique that's an alternative, if you like. It's much newer. It's from 2006, from Eric Betzig from Harvard. And the principle here is, again, that you have your fluorescent markers, but this time you exploit the possibility that they can be turned off, on or off. So what you do, you excite your whole thing as before with a blue laser, but then you have this switching laser. And the switching laser, you use a very low intensity, and that means that you have a very low probability for each fluorophore to be switched on. So in effect, most fluorophores stay in their off state, only very few enter the on state, and what you see is, in this case, 2 of these fluorophores have been switched on, and you see 2 green blobs over here. But then you know, under each blob, there is with very high probability only exactly 1 fluorophore, and you can localize that very precisely. And then you do that many times, you aggregate the data, and then you get an image, super resolution image. Okay, and the third method I would like to introduce is based on the fact that each molecule has also an orientation. So it's got a dipole vector. And light also has an orientation. It's called the polarization. And the photon yield of this fluorophore is maximal if the dipole moment and the polarization are aligned. And it's sort of a continuous function of the angle between the two. So if we rotate the polarization of the light, we can make them, these molecules, blink in time, if you like. And if each molecule has a random orientation, that means each molecule has a random blinking pattern. So this is what you do. You use your excitation laser, but this time with a polarization rotator in the middle. And then what you observe is this such a sequence of images. One may look like this, another one like this, and another one like this. And the task is then to infer the distribution of molecules underlying that. So these are 3 methods that can be used for super resolution. And the basic principle that unites them is sparsity. The principle by which you can break the diffraction limit is that you cause, by whatever means you have, to light up the molecules not all at the same time, but at different times or with different phases. So in this example here, if you have 2 molecules close by and they light up at the same time, you get something like this, and you have no idea what's going on. Is it 1? Is it 2? Is it 3? Where are they? You don't know. But if you can separate them, so at the one time you only see this, then you know, okay, it'll be there in the middle. And the other one, it will be there in the middle. So you can achieve super resolution. Now, how do we go about and model this? What we want to know is this function f, concentration of the fluorescent dye. But we don't want, or we can't, we observe that directly. What we observe is the function g, which is the concentration of excitable molecules at a time t. So we are changing, so that's the sparsification that I was talking about. And then we've got this point spread function, and we are convolving this function g with a point spread function, and that is the average of what we would observe, and that enters this, for example, Poisson process as an inhomogeneous parameter. And that's then is our image model. Yes?
These are methods to understand quality, to track or understand the content or the number of proteins in a region. It's not to figure out the structure of the proteins themselves, right?
That's right, exactly, yes. Because you don't see the proteins, you only see the fluorescent marker which is attached to them. So you have no means of seeing the structure of proteins. Yeah.
How do you know that you were able to attach a fluorescent dye to all proteins of interest?
One method is to modify that genetically, so it just is expressed that way. There are other methods, I think, but I'm not an expert on that. So for this step procedure, what you do is you have your concentration F, and you multiply it with a sharply peaked function S, which is centered around 0, but then you shift it along a path, that's this scanning path axis naught there, and you scan the whole image, and then that's what you observe. And for single marker switching, it's different, but in this formalism, it looks quite similar. You have your concentration F, and you multiply it with a random process, which is sparse. So you've only very few locations where you can actually see anything. And so because this G is now sparse in X at every point in time, this statistical inverse problem to get from your observed image to the actual G is a well-posed problem. And once you solve that, and that's fairly straightforward, you can just add up all points in time that you have, and that gives you an estimate of your underlying density.
So, and what do you know in these two problems? Do you know S and do you know E?
Yes, you know S, you know X naught. You don't know E, but it doesn't matter, because maybe I will show in a minute. And now for the third method, it's again quite similar, but this time your concentration is multiplied with a time-dependent function. So D is a time-dependent function. It's periodic because you're rotating your polarization periodically. And each molecule has a random orientation and therefore a random phase shift tau. So if you put all this in, you can write your function G as a convolution of a density in the phase shift domain with your modulation function D. And the point now is that H doesn't have to be sparse in X, but by construction, it's sparse in the phase shift domain. It's sparse in theta. So again, you have created some sparsity, and you can use that to break the resolution limit. Okay, now I'd like to show what kind of methods we can use and what challenges arise in this context. So here's an example at first of a STED image. So this is a sort of conventional microscopy, and this is the same thing, exactly the same sample, but with STED. And I think you can see that the resolution increase is really substantial. It's a lot better. This is of a protein called vimentin in a cell. But this image, although it's very much better than this one, still isn't the real thing. It's not the real concentration of the fluorescent dye, because there's still noise on it, quite a lot of noise, in fact, because we are observing very few photons. And there's also still some sort of blur on it. There's still a point spread function at work, but it's much smaller, of course, but still. So we can try to improve that, and one way to do that is to use multiscale constraint optimization. So what do you do there? You take a set of subsets of the image, for example, a set of all rectangles or of all squares or whatever seems appropriate, and you choose a prior functional that encodes your prior knowledge about your density. And it can be the TV norm, L1 norm, L2 of gradient, or many other things. And then on each of your chosen rectangles, for example, you perform a test, a statistical test, whether the image you observe is compatible with a candidate function f that you choose. So if this test is fulfilled, then your candidate function is in the feasible set. And from all those functions in the feasible set, you choose that one which minimizes your prior functional. And this is a way of constructing an estimate of your underlying density, f hat. And it's some advantages, because what you need is these statistical tests, but that's usually easy to construct for Gaussian statistics or Poisson or many other things. And what you need to know is the 1 minus alpha quantile of this statistic, but that can be calculated in many cases, or even if it cannot be calculated, can be simulated on test images. So you can do this very easily, this testing. And this quantile, this alpha here, is the only parameter in this whole scheme. And that's very attractive, because there's one only overall parameter, and that has a very clear statistical interpretation, because it gives us a smoothness guarantee. The smoothness guarantee is that the estimator is at least as smooth as the real function in terms of j, with a probability at least 1 minus alpha. So that's gives us some statistical guarantees about our estimator. And here's some examples.
What are those example functions? What do they look like?
How do you mean example functions?
I mean that set you're trying to choose from.
For example, for the next image here, I chose the set of all rectangles of size 1 by 1 up to 20 by 20. So, but you can make many other choices, and they have a big influence, or they can have a big influence. Yeah, that's certainly true. So this is now this little region up there, and this is the original, very blurred, very noisy. This is a reconstructed version, and you can see some details in this region, for example, which you cannot see here at all. And this is for the confocal, so the standard microscopic image, but we can do the same for the stat image. It's the same region. And here you can verify that the details that I found in the previous image are actually compatible with the real thing that's going on underneath. And again, we can do this same thing for this stat images and get a statistically valid estimate of our function f. Okay. Now let's switch to this other kind of super resolution method, the single marker switching, where you switch on the molecules one by one. And here's an example image. So I think this is, refers to your question. This is the sum of 40 frames, in fact. So each individual frame is much sparser than this one. But you don't need to know this function E, because you can identify these little spots in each image. You can just see what it looks like. I don't know if I'm making myself clear, but what you see, whenever you see a spot of light there, you know there must be one molecule underneath of that. And you can automate this and localize the molecules, and that is what this picture then looks like. This is for the first 40 frames up here. And then this is for if you add all frames together. So this gives a very blurry image, sort of a standard microscopic image. But if you do the localization first and then add, you get a very sharp image of the whole thing. Well, that's quite an ingenious method. But as I think you remarked, it may very well be the case that over the course of time, the whole image moves, because it takes of the order of minutes, I think, for such an image to be taken. So, and these yellow markers here, they show an actual drift over this time. So, in fact, we would like to correct for that. And one way of doing that is to put in these fiducial markers, so you can track them, you know the drift, and you can correct for that. But even nicer would be if you could do that from the data itself without any markers. And that's this drift estimation, where you have a parametric drift model, and you put that into your fluorophore here as a time-dependent drift. And then you can formulate a certain contrast functional. And if you minimize that functional with respect to your drift parameters, then you get an estimated drift, and you can use that to correct your images. And that gives very accurate results, as you can see in this image. So here on the top row is the uncorrected image, and corrected with the fiducial markers, and corrected just from the data alone. And you can see that fiducial markers and data alone gives both very good results. And this is the original if you don't do the correction. Okay. Now I'll almost finished. Now for spot, I said that we have sparsity in the phase shift domain. So what we do here, for example, is we write down a likelihood term for our image sequence that we record, and we put in a sparsity enforcing penalty there at the end. And then we can try to minimize this, and that gives us an estimator of our density in phase shift space. And this is, in fact, a convex optimization problem with non-smooth terms, because you've got the L1 norm there. So it can be done with a number of methods. We've got this software available, which is based on the back-to-bool algorithm, but you can also use Chambol-POC or ADMM or other methods. And with that, you can then reconstruct your image. And this is, on the left, the original. This is the resolution-enhanced image. And here on the right, you see, if I switch through it, phase slices. So at each phase slice, you get a different image. And if you superpose all of them, you get the image in the middle. And in fact, what I said in the beginning, there's a lot of blur going on here. But in this resolution-enhanced image, you can actually see that this is there's some structure here at the end of this spine head. Okay, and with that, I'm finished with my overview over the statistical methods and problems in super-resolution microscopy. So what I would like to say is that all of this requires you to take into account the single molecules and the single individual photons that you are observing. And to do that, you need a detailed physical, chemical, statistical description of the underlying processes that are going on. And you need to put all that into your methods. And then, on top of that, you have to apply advanced statistical methods in order to get the optimal image from out of there, including possibly confidence statements. And all these problems can be formulated as non-smooth convex optimization problems on a large scale. So it's really a tough problem to do numerically. Okay, thank you very much.
So that was really interesting. So even if you get this very, very clear kind of point estimate of this function or protein or whatever you're doing, is that the end goal of the analysis, or do you actually want to do some sort of analysis of that image? I think that would be very, very challenging. But
I think
you're just trying to give them a nice picture for their paper, or you have many of these pictures and you want to
ideally
do something with the picture.
Yeah, I mean, ideally you would like to have an estimate of your underlying distribution, concentration, and have some confidence statements about that, so you can make whatever biological statements you would like to make. And I think that's the biological statements I can't do. Those is for the biologists. But I would like to be able to make sort of confidence statements about how good is the image that I'm giving you. That's
but they're looking for some feature.
They are, yeah, sure, yeah.
I mean, that's essentially the. I would say nowadays one of the major tools in medical and biological research. These kinds of things, right? I mean, there are tons of tasks. For example, they succeeded now in Planck. I think just about a year ago, if I remember correctly, really to see neurons live firing. So this is a very important issue here, which should maybe emphasized again. We talk about living cell microscopy. So you really can see transport processes, communication processes, also, for example, in detecting cancer, you can see in certain parts of the cells which kind of processes are now going to wrong directions, where anomalies now start to exist and so on. But that's a huge area. So it's not just to make a few of nice artwork images. And of course, the second question I think you had is if you think, for example, about processing lots of these images, a lot of things, of course, now go more and more automatically, and it certainly will go to the direction that we will have just huge, large sequences of these images which have to be processed in a more automatic way, and that would be, in a sense, the first pre-processing step to give you as accurate as possible reconstruction of the history. So let's thank you again, and our next speaker.
Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.